I'm Dr. Chris Masterjohn of chrismasterjohnphd.com, and I'm here with Alex Leaf.
Hey.
And you are listening to episode 66 of Mastering Nutrition, where we talk about pantothenic acid, part 2, foods, lab tests, and supplements.
This is Mastering Nutrition with Chris Masterjohn. Take control of your health, master the science, and apply it like a pro. Are you ready?
This is part 2 of our 2-part series on pantothenic acid or vitamin B5, where I'm covering the basic science, how much we need from food, how to get it from food, how to use lab testing to know if we have enough, and Alex is covering the health outcomes and other effects of supplements. If you missed part 1, you can find it at chrismasterjohnphd.com/64, and because this is episode 66, you'll be able to find the show notes for this episode at chrismasterjohnphd.com/66. To catch you up to speed and review the signs and symptoms of vitamin B5 deficiency are fatigue, loss of enthusiasm and energy, insomnia, grumpy mood, cramps, including but not limited to abdominal cramps, gut problems like increased farting, feeling nauseated, sometimes throwing up, getting sick more often, a high pulse in response to minor exertion, and potentially neurological problems causing tingling or numbness in the hands and feet, and even stranger things like an inability to walk on tiptoe, and occasionally manifesting in individuals in inconsistent patterns that might cause constant burning sensations or constant numbness in the hands or feet and walking funny by lifting up the foot more than you think you should normally have to when you walk. The forms of vitamin B5 that we use for enzymatic cofactors to support our metabolism are 4-phosphopantothene and coenzyme A. When we eat food, 85% of the vitamin B5 is found as 4-phosphopantothene and coenzyme A. Coenzyme A is a complicated molecule that takes a while to digest, and the 4-phosphopantothene is all bound to proteins, which requires digesting through the protein to get to it. And as a consequence, only about 50% of the vitamin B5 in food is fully digested and absorbed. Contrary to the claims of major authoritative reviews, we do absorb, according to the evidence, significant amounts of forms of B5 from food besides just pantothenic acid. So it is claimed that we digest it all down to pantothenic acid, which is found in the free form in about 15% of food vitamin B5 is pantothenic acid. And pantothenic acid is not something that we use in the body apart from converting it to 4-phosphopantothene and coenzyme A in a rather complicated and very energy intensive and arguably difficult process that involves not just magnesium and ATP, but also involves cysteine, which we need vitamin B6 and glycine to get, as well as sufficient protein, including the sulfur amino acids that are more abundant in animal protein than plant protein. So it is not a simple task to make the conversion of pantothenic acid to 4-phosphopantothene and to coenzyme A. And in fact, there are various genetic defects causing severe problems in the ability to make those conversions. But if you go back and look at the old literature, actually it indicates that we don't digest it all down to pantothenic acid, but we do absorb 4-phosphopantothene and pantothene, both of which are more easily converted to coenzyme A or to 4-phosphopantothene itself, which we absorb itself, the other major enzymatic cofactor that we need. When we absorb B5 from food or supplements, it does not stay in our blood for long. It's very quickly transported to our other tissues. It's known that pantothenic acid is not a major circulating form of vitamin B5. It's maybe 3 to 8% of the total. And until recently, we didn't really know what the other forms were, but there's recent data suggesting that the main circulating form delivered to tissues may be 4-phosphopantothene. We have very important needs to regulate the amount of coenzyme A that is free and the amount of coenzyme A that is bound to other molecules. And as a result of that need, most likely, we break down a small amount every day into pantothenic acid. We don't know what the total body stores are, but they might be about 4.5 grams, and we may be excreting in the urine around 4 milligrams or varying depending on total body status. And I'll make the argument in this episode that, in fact, urinary concentrations are very useful reflectors of whole body nutritional status for vitamin B5, and blood concentrations are not. During adolescence, there is probably a markedly increased need for B5 to produce sex hormones. And one hypothesis is that this is why we have an increased risk of acne during the teenage years. During pregnancy, and especially in the third trimester, there's active movement of B5 to the fetus at the mother's expense. And during lactation, there is a movement of significant amounts of pantothenic acid into the milk at the mother's expense. Doses of supplements have been used at grams per day, and they appear to be safe and to be used with high efficiency. This is probably a matter of repleting the whole body stores, which, as I mentioned, greatly exceed our daily intakes. In this episode, we talk about what is the ideal marker for nutritional status. What can we do with that to learn how much we need? How do we get that from food? Do we, in fact, get any from the microbiome? What are some of the factors that would make us need more? What, if anything at all, can we say about the prevalence of B5 deficiency? Alex goes in great detail into talking about the differences between pantothine, pantothenate, and dexpantothal. He talks about oral and topical uses of B5 supplements. He talks about using them for blood lipids, for acne, and for wound healing, and addresses the question of whether B5 can influence exercise performance or whether exercise performance influences our need for B5 and whether it can do anything for the graying of hair or for arthritis. We talk about dosages, we talk about safety, and we talk about how dosages and safety differ between the different forms. And we talk about things like whether you should take it with food, whether you should spread it out over the day or take it all at once, and practical things of that nature. Again, this is part 2 of a 2-part series. You can find the other part at chrismasterjohnphd.com/64. This is part of a larger series on managing nutritional status, and you can find all the episodes at chrismasterjohnphd.com/marker #episodes. You can get a transcript if you sign up for the CMJ Masterpass at chrismasterjohnphd.com/masterpass/mastering nutrition, which that specific URL will give you a 10% lifetime discount. And you can get all these links in the description of this episode. So let's dig right into all the science right after this word from my sponsors. This episode is brought to you by Ancestral Supplements. Traditional peoples, Native Americans, and early ancestral healers believed that eating the organs from a healthy animal would strengthen and support the health of the corresponding organ of the individual. For example, the traditional way of treating a person with a weak heart was to feed the person the heart of a healthy animal. Modern science makes sense of this. Heart is uniquely rich in coenzyme Q10, which supports heart health. The importance of eating organs, though, is much broader than simply matching the organ you eat to the organ you want to nourish. For example, natives of the Arctic had very limited access to plant foods and got their vitamin C from adrenal glands. And vitamin C is important to far more parts of your body than simply your adrenals. In his epic work, Nutrition and Physical Degeneration, Weston Price recorded a story of natives who cured blindness using eyeballs, which are very rich in vitamin A. But now that we understand vitamin A, we know that we can get even more vitamin A by eating liver, making liver good for your eyes. Our ancestors made liberal use of organ meats both to be economical and to utilize their healing and nourishing properties. Animals in the wild do the same. Weston Price had also recorded a story of how the zoos in his era were capturing lions, tigers, and leopards, oh my, only to watch them become infertile in captivity. Researchers then observed what the lions did when they killed zebras in the wild. What they did was they went straight for the organs and bone marrow, leaving the muscle meat behind for the birds. But even the birds took what they could of the organs and bone marrow. Price reported that once the zookeepers started feeding the animals organ meats, boom, their fertility returned. The problem I often encounter, though, is that many people just don't like eating organ meats. Let's face it, if you weren't raised on them, it can be very hard to acquire a taste for them. That is where Ancestral comes in. Ancestral Supplements has a nose-to-tail product line of grass-fed liver, organ meats, living collagen, bone marrow, and more, all in the convenience of a gelatin capsule. For more information or to buy any of their products, go to ancestralsupplements.com. Ancestral Supplements, putting back in what the modern world has left out. This episode is brought to you by Ample. Ample is incredible. It's a meal in a bottle that takes a total of 2 minutes to prepare, consume, and clean up. 2 minutes. I'm not kidding. Now, I know what you're thinking. Anything that quick just has to be made of synthetic ingredients that you'd have a hard time pronouncing and wouldn't want to put into your body. But it's not. Ample is made entirely from natural ingredients and designed to provide an optimal balance between protein, fat, and carbs, as well as all the vitamins and minerals that you'd need in a single meal. There's no question that it's always best to sit down and take your time eating a home-cooked meal from fresh ingredients. But let's face it, oftentimes we just don't have time for that. If you live a busy life like I do and your goal is to get things done, you need quality fuel that you can get into your system quickly. Here's a great example where Ample is perfect for me. When I shoot videos, it takes hours to set up and break down all of my equipment. So I try to get as many videos shot in a day as possible. This prevents wasting a lot of time on setup and helps me conserve big blocks of time outside of shooting videos to get into a flow state where I can research something to my heart's content and spend all the time I need thinking about it creatively and analytically. But if I spend hours dealing with recording equipment plus hours spent preparing food, eating it, and cleaning up, there's no time left over to actually shoot any videos. So on recording days, I use Ample to maximize efficiency and focus on getting things done. Ample comes in 3 versions: original, keto, and vegan. And each version comes in 2 portion sizes, 400 calorie and 600 calorie. The 600 calorie original version gives me 37 grams of protein from a mix of grass-fed whey and collagen, which promotes satiety and flips my brain on. Its fat comes from coconut oil and macadamia nut oil. I like these oils because they're low in polyunsaturated fatty acids, or PUFAs, oils that promote aging and are usually loaded into the processed foods that most people eat when they need something on the go. The coconut oil provides some medium-chain fats to keep my energy levels up, too. The carbs, the vitamins, and the minerals all come exclusively from food sources like sweet potatoes, bananas, cocoa powder, wheat and barley grass, and chlorella. It's full of natural prebiotic fibers and probiotics to promote a healthy microbiome, and the gentle sweetness comes from a mix of honey, monk fruit, and stevia. I just mix it with water, drink it, rinse out the bottle, and boom, 2 minutes in, and I'm fully fueled and ready to face the next phase of the day. I first came across Ample when I met its founder and CEO, Connor Young, at PaleoFX a few years ago. Connor inspired me with his vision for Ample, which I anticipate will be much more than a meal in a bottle in the near future. I've become an official advisor to Ample, and I'll be helping Ample design scientific research that will lead both to an ever-improving Ample and, I hope, meaningful contributions to our understanding of how to use nutrition to help people be healthier and happier and perform better at the challenges that they care most about. As a listener to the Mastering Nutrition podcast, I've also worked out a special deal for you. If you use the discount code CHRIS15, you'll get 15% off your first order of Ample. To get your discount, go to amplemeal.com. That's amplemeal.com, 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 and use the code CHRIS15 at checkout. Mouse models to address it. And in the mouse, you don't have a perfect model because in the human, there's PANK1, which is the enzyme that's dominant in the liver, and PANK2 dominant in the nervous system. And the genetic defect is just in PANK2. And in the mice, it's not divided the same way. Their PANK1 is most abundant in liver, but their PANK2 is most abundant in their testes rather than their brain. So there's no specific way to target the defect right into the brain in mice like there is in humans. But as I mentioned before at the beginning, when the PANK2 defective mice are given a ketogenic diet, they get dramatically worse. They really start with devastating neurodegeneration. And pantothenic supplementation, the form that Alex was talking about, can rescue them. Now, in humans, many people have tried a ketogenic diet, and in net, no one's reporting anything net positive or devastating or harmful from the ketogenic diet. So basically, ketogenic diets so far anecdotally don't seem to be doing anything one way or the other for this disorder. Also in humans, there are a lot of people trying pantothenic supplements, but it's not clear whether they're working. There's no clinical trials to gauge the progress, and there's no overwhelming anecdotal support for using pantothenic versus pantothenic. However, one thing to note is that pantothenic generates 2 molecules of pantothenic when it's split, and pantothenic does have some of the ability to bypass some of the steps and some of the ATP utilization that the 4-phosphopantothenic and the coenzyme A would in food. It's just that pantothenic is not going to bypass anywhere near as much as you would bypass by getting 4-phosphopantothenic from food and from the digestion of coenzyme A in food. So researchers have continued to try to find things to do about this. And this was the basis for why in 2015 they found that 4-phosphopantothenic was stable in blood, and they tried it in mouse models and fruit fly models of the pantothenic kinase deficiency. And it works. The problem is that 4-phosphopantothenic is very difficult to synthesize in high amounts, in high purity, to give it to people as a high-dose supplement or as a drug, depending on how you would view supplementation with it. Also, it has a free sulfur group that makes it have a very bad shelf life. Now, if you compare that to the 4-phosphopantothenic in food, you don't have to synthesize it because the animals and plants are doing it for you. And you don't have to worry about the stability because it's bound up in the proteins in that food that protect it until you digest it. So when you're talking about food 4-phosphopantothenic, you don't have the stability issues that you have with developing it in pure form. So what they did was they developed an acetylated form of it to protect the sulfur from oxidation. And what they found was that in human embryonic kidney cells and in fruit fly larvae and in mice, when you made them genetically defective in pantothenic kinase, you could completely rescue the disease by feeding the acetylated form of 4-phosphopantothenic. Remember, 4-phosphopantothenic is one of the major forms in food. What's the other major form in food? Coenzyme A. What else can rescue fruit flies, larvae, live fruit flies, mice, zebrafish, and human cell models of pantothenic kinase deficiency? Coenzyme A. In every single model, preformed coenzyme A completely rescues the phenotype. The genetic disorder becomes not a genetic disorder when provided with enough 4-phosphopantothenic or coenzyme A, which are the 85% of the B5 found in food. And that makes you wonder what kind of doses do you need? Because as we'll talk about in part 2, there's not a lot of pantothenic in food, and really you're looking at nutritional yeast having the most and liver being a distant second and everything else being way below that. Which is funny, right? Because pantothenic acid is named after the fact that it is everywhere and in everything, but it's not very evenly distributed in the food supply. So I wonder what would happen with nutritional yeast supplementation and liver, eating a lot of liver, or, at least managed, depending on the doses they need. You could overdose on the vitamin A or copper in liver. You could overdose on the selenium in nutritional yeast. But I do wonder whether the food forms of vitamin B5 from the foods that no one eats could have a significant benefit here. Now, there are also genetic defects in the final steps of coenzyme A synthesis and in some of the ones in the middle that are more recently discovered and that are more rarer. And for the final steps, none of the models have shown any benefit to coenzyme A or 4-phosphopantothene. And the way the disease manifests is very similar. So there are people with coenzyme A genetic defects who probably cannot be rescued by the supply of 4-phosphopantothene or any of these other things, although maybe certain drugs will be developed in order to help them. But where the benefit really is, where it's very promising, is in people with the pantothenic kinase deficiency. And so this has to make you wonder, what if there are polymorphisms that result in a 10 or 20% loss of pantothenic kinase activity? Could there be many of us out there who would be completely fine if we had enough food form vitamin B5, but who would suffer from at least moderate problems if we didn't, and who would not benefit from taking pantothenic acid supplements the way we would benefit from food form B5? I think you can make a very compelling case from this that consuming B5 in food is at least insurance against the possibility that you could have problems converting the pantothenic acid in supplements. And I think you could make a hypothesis that there may be polymorphisms out there in the world right now that are making some people benefit tremendously from eating the foods that are very rich in vitamin B5. So with that, this is the end of part 1, but we will come back for part 2 to talk about markers of nutritional status, how much we need, how to get it from food, and how to get it from supplements. All right, so let's talk about vitamin B5 and what we know about how much we need. First, we need to understand what we should be using as markers for nutritional status. I will make the case that the best marker in fact, the only legitimate marker of nutritional status is urine concentrations, and that blood concentrations are not a useful marker of nutritional status. In fact, I'm not sure exactly what I'm going to do for my testing nutritional status, the ultimate cheat sheet. I am going to have to remove the recommendation to use blood levels of pantothenic acid that are offered by LabCorp, but the only lab that offers urine testing for pantothenic acid is the Great Plains Organic Acids test, and you have to order the whole test for a lot of money in order to get that. So I think what I will do is suggest that people who have signs and symptoms that seem like they could be B5 deficiency should use the Great Plains test instead of the Genova test when doing comprehensive nutritional screening. But that people with financial limitations should just try, because of its safety, ways of increasing vitamin B5 intake through foods and supplements if they believe that they might be deficient. So let's lay out that case that that urine and not blood is where you want to measure it. In the study by Peggy Fry in 1976 that I mentioned at the beginning of the last episode, where people were put on 9 weeks of either a pantothenic acid deficient synthetic diet or a pantothenic acid replete synthetic diet, they looked at the urinary excretion and the blood concentrations over many periods in this study. And what you find is that in the urine, the pantothenic acid in the deficient group dropped each week of the study, getting worse and worse and worse. By the end of the study, dropping 74%. In the replete group, they were actually getting better pantothenic than what was in their diet, because their diet was, probably mediocre. And then now they're getting a synthetic diet with 10 milligrams per day. And the supplement is actually probably absorbed better than whatever was in the diet, because supplements are probably absorbed at low doses with close to 100% efficiency, and diet is absorbed with about 50%. So they have better pantothenate status than they came into the study, and their urine concentration goes up each week of the study, by the end of it going up 47%. Now, contrast this with what happened in the blood. In the blood, in the deficient group, it dropped by 28%, but then it started recovering. So instead of getting worse and worse and worse through the course of the study, after an initial drop, it started getting better and better and better by the end of the study. In the replete group, even though they were getting better pantothenic acid status, they actually dropped 12% at first and then fully recovered by the end. So part of the drop in pantothenic acid early in the study, even in the deficient group, was something about the synthetic diet that they put them on, because it happened in the control group as well. And what you find in both cases is that the blood tends to recover over time. What if they had put these people on a deficient diet for 18 weeks? Probably the blood would have gone back up to what it had been in the first place. So in dramatic contrast to the blood results, the urine results faithfully reflect the degree of deficiency and the length of time that you've been deficient for. Another thing that's really interesting is that they took both groups at the end of the study. And they said, okay, one group has been deficient for 9 weeks. The other group has been really good status for 9 weeks by taking 10 milligrams a day. Now let's give you all 100 milligrams per day. What happened in the group that had been getting 10 milligrams per day for the 9 weeks, the control group, they immediately shot up to from 5.8 milligrams of pantothenic acid in their urine per day to 52, and then to 62. So they shot up very fast. In the deficient group, they shot up fast, but only to 36. It took them a whole week to get up to 59. So after 1 week of taking 100 milligrams a day, everyone gets up to around 60. But the group that had been deficient in the past took a lot longer to get their urine concentrations up that high. Then they let everybody, just start eating what they usually eat. So everyone back to regular diets that were mediocre. The deficient group, the group that had been deficient, this is now over a week since they had been in the deficiency group, right? So these people, 9 weeks of deficiency, 1 week of 100 milligrams a day, and now they go back to their regular diet for a week. So it's been 2 weeks since they were part of the deficiency group. Their urinary concentrations go down from 60 to 13. But in the sufficient group, who had been getting good pantothenate dosing all along, they went up to 100 milligrams a day for a week. They dropped down to their usual diet for a week. They only went down to 20. So the past experience of sufficiency led to 20% higher urine values at the very end of the study compared to the past experience of deficiency. So what's going on in this? I believe that you lose some certain percentage of your total body stores of pantothenic acid on a daily basis. And this is because you're trying very hard to conserve your coenzyme A and your 4-phosphopantothene, but if you need to regulate the acetyl-CoA to free-CoA ratio, or you need to regulate the total CoA pool, you're going to wind up degrading some of it. And because you're always going through a fasting feeding cycle, you're always shifting around the CoA needs of more need in the liver during the fasting state, for example, where the liver is acting as a metabolic hub to support the energy needs of the rest of the body by making glucose and by making ketones. So as you're always cycling through the fasting feeding cycle, you're shifting the CoA needs around to the different tissues, and they're always going to have some that's being degraded as a result of that. That's the model that I'm using to understand why you're losing this little bit in the urine every day. But just like, say, creatine, you have 120 grams of creatine in your body, and you lose about 2 grams a day, which is 1.6%. And in that case, it's because the creatine gets just randomly damaged. It's not a controlled process. But if you look at the excretion of creatinine in the urine, which is a breakdown product of creatine, it's proportional to the total creatine stores of your body. So if you increase your muscle mass, your urinary creatinine is going to go up. If you start taking creatine and increase the amount of creatine stored in your muscles, your urinary creatinine is going to go up. I believe that urinary pantothenic acid is very much the same thing. If you increase your whole body stores, then there's some small percentage of the whole body stores that will be degraded, and that is what is reflected in the urine. That's why urine faithfully goes up when your status gets better and better. Urine faithfully goes down when your status gets worse and worse. And that's why when you see Peggy Fry's study in 1976 that I was just describing, why the past history of deficiency or sufficiency is reflected so clearly in the urine stores. And by contrast, what is blood reflecting? 90% is in the erythrocytes, the red blood cells. And what is it serving? It's serving the metabolic needs of the erythrocytes. And so the erythrocytes, their needs don't change because you're deficient. Their needs don't change because you're sufficient. Their needs don't change because you're megadosing. And so their needs, as long as you're not pushing the system to dramatic levels one way or the other, their needs are going to stay pretty constant. The blood values are going to stay pretty regulated. Now, it gets a little worse. So Eisenstadt in 1986 looked at blood levels and urine levels and intake. And they found that intake correlated 14% with the levels in blood and 36% in the urine. So at first you look at this and you say, okay, it correlates 3 times better with urine than with blood. But there's another important point. The urine didn't correlate with the blood at all. The correlation between urine and blood was 0. And so what that means is that you have to pick one. If both of them were offering some information about status, then whatever that information about status is that's contributing to either of them would make blood and urine correlate. But if they don't correlate at all, only one of them can be telling us about status. And if only one of them is telling us about status, we have to pick one. And if we pick one, we have to take the thing that we know reflects long-term status, that we know faithfully gets worse when you get worse, that faithfully gets better when you get better. And that one is not blood, it's urine. In further support of this, Fox and Linkswiller in 1961 took women on an average diet and put them on 3 doses of Pantothenic. There were doses that were lower and doses that were higher than what they were usually taking in. In the low dose, where they dropped to a lower Pantothenic intake, they were eating 2.8 milligrams per day, and they were losing somewhere between 3.2 and 4 milligrams per day in their urine. If urine was going to reflect acute intake, that would be impossible. They're not urinating out the excess over what they're taking in. They're urinating out something that's reflecting their full body stores. That's why they're urinating out more than what they took in, because they were only recently put on the deficient diet and their body stores are intermediate or maybe even pretty decent. And so the amount that they lose in their urine has nothing to do with this new deficient diet they're on. It has everything to do with their full body stores. In further support, in late pregnancy, Pantothenic levels drop in blood, but they don't drop in urine. And milk correlates with diet and with urine, but not blood. Supplementation in pregnancy raises milk levels 5-fold, but not blood. In further support, in pregnant women, the blood concentrations drop as the Pantothenic moves towards the fetus through the umbilical cord, but the urine doesn't drop. And that's because the women, if they are well nourished during pregnancy, still have good whole body stores. Their urine reflects their whole body stores, and their blood drops, reflecting of what they are doing with it right now, because they are transporting it from their blood into the umbilical cord blood. And then in lactating mothers, the milk correlates very well with diet and with urine, but not with blood. This reflects the fact that you're putting pantothenate into the milk in reflections of your ability to do so, which are reflected in your whole body stores, and that your urine is a faithful reflection of your whole body stores. But you don't see the correlation in blood, because what you're immediately doing is using your blood to transport the pantothenate into the milk. So the blood is reflecting very acute events of where things are going where, and it's reflecting the needs of the erythrocytes. Whereas the urine, as long as you're not currently taking a megadose, I believe, will faithfully reflect the whole body stores. If you take a megadose of a supplement, some portion of that that's not retained does go in the urine. But when you go off that supplement for a day, your urine goes back to reflecting your whole body stores. So can we use this to determine how much we need? In the 1998 DRI report, they used the fact that in 19 to 50-year-old adults, urinary excretion equaled intake at 4 milligrams per day. And they basically used the idea that you want to maintain pantothenate status balance. You don't want to urinate out more than you're taking in. So they looked at data and they said, okay, the point at which your intake equals your excretion is at 4 milligrams per day. So let's say that 5 milligrams per day is enough to make sure that you're not losing pantothenate over time. And then they extrapolated downwards for all the children based on body weight values. For infants 0 to 6 months, they took the average amount that they were taking in from milk. And for pregnant women, they rounded the data, the average consumption among pregnant women, up to 6 milligrams. And because there's an extra 1.7 milligrams put into the milk, they increased the adequate intake to 7 milligrams for nursing mothers. Let's come back to this idea of pantothenic acid balance. How much can we trust the concept of not wanting to take in less pantothenic acid than we are excreting in the urine? For that, Alex, I know you've done a lot of research in using nitrogen balance as a marker of protein needs, which is a very similar idea. Can you comment on, what you've learned about the limitations of using nitrogen balance for protein as an analogy?
Yeah, it all comes back to the body being smart and adaptable and not trying to actively kill itself. So with nitrogen balance in protein studies, the issue is that they're waiting for the body to establish an equilibrium at various intake levels of protein. And this requires being on, for example, a low protein diet for 1 to 2 weeks. But the body is smart, and that's adequate time for it to start down-regulating important processes that are not essential for survival, but that do contribute to overall health, such as immune function and muscle protein synthesis. And so nitrogen balance studies are inherently flawed because they provide a lower limit of what's necessary for survival, but not what's necessary to maximize health over the long term. If we're going to bring it back to pantothenic acid, then we can presume it would be similar, where the urinary excretion would be, the body could adapt and therefore reach an equilibrium based on survival needs rather than optimal health needs.
Yeah. So the first idea here is that there is no rationale in the DRI report for using the balance approach. And as Alex pointed out, we know very well that the body engages in all kinds of compensations to try to preserve itself when you go on low intakes of something. But there's a bigger problem here, which is that if there's a math problem here, if dietary pantothenate is 50% bioavailable, then the people who are consuming 4 milligrams a day are absorbing 2 milligrams a day. And if an intake of 4 milligrams a day equals a urinary excretion of 4 milligrams a day, then they're actually absorbing 2 milligrams a day and losing 4. So they're losing 2 milligrams a day. Now, we don't know how big the B5 pool is in humans. I made a rough guesstimate based on rat data, adjusting for size of organ weights, that it would be 4.5 grams. And Alex discussed some study in episode 1 suggesting that we can actually retain 5.6 grams of a megadose. So we don't know exactly what the pool is, and we don't know how variable it is. But if we just slap some numbers on this, if you're consuming 4 milligrams a day, you're absorbing 2 milligrams a day, and you're losing 4, you have a net loss of 2 milligrams a day, you would run out of your whole body stores in 6 years if your whole body stores are 4.5 grams. Obviously, that's not going to happen, and you're going to engage in all kinds of compensations to preserve the B5 pool, including urinating less out of it. But that doesn't mean that you're not getting very far away from your optimal health level. And I think it's very, very interesting to take this back to the original studies of deficiency in the 1950s. They were giving the people a pantothenic acid deficient diet and an antagonist. And 6 weeks through the study, several weeks through the study, they had to increase the dose of the antagonist because the people were adapting and conserving and stuff. But they were still clearly not that healthy. And we can also say from the Peggy Fry study in the 1970s that 9 weeks just produced fatigue and loss of enthusiasm and didn't produce the worst neurological effects. Well, why? Because over those 9 weeks, what happened to the urinary pantothenic acid? It shot down because they were conserving their stores, right? So it seems like at 4 milligrams a day, we're probably not even in pantothenic acid balance. And we can be pretty sure that that's actually deficient. But we don't really know, what is the potential to optimize health with higher intakes. And one thing that I think is worth asking is, if we just use our imagination for a moment, I'd like to think about how much might we be able to improve our pantothenic acid pools over time with a very good diet from the average. And we don't have data on that, but Alex, can you come in and talk about, as an analogy with muscle mass, there's an average that we have from just doing normal life. What is our potential to expand that pool of muscle protein?
So if we go back to the reference man that was put together in 1975, weighing 70 kilograms or about 154 pounds, they have 40% of their body weight as muscle mass, which is about 62 pounds. And when we look at estimates of how much muscle you can put on with consistent resistance training as we work towards our maximum genetic potential, we can see that you can put on 20 to 25 pounds in about 1 year, which corresponds to 2 pounds per month. After that 1 year, during the second year, that rate slows by about half, and it slows by another half during the third year of consistent proper training. But overall, over a 3-year period of consistent proper resistance training, you can expect for that reference man with 62 pounds of muscle, he could expect to double his muscle mass before it begins slowing down to, being only about 2 to 3 pounds of muscle per year, if that, where someone would be considered a very advanced trainee. But as we discussed in part 1 of this podcast, with most of the pantothenic acid being stored in skeletal muscle tissue, 30 to 40% of it, and we can expect from all of the studies in humans that these are done in being just lightly active, certainly not regularly resistance training people, that they could double their biggest storage site of pantothenic acid. So there is a lot of potential to expand this pool.
Yeah, I mean, that's pretty fascinating, because if you double your muscle mass, you almost certainly need a lot more pantothenic acid, because just to maintain the normal concentrations in that muscle pool. But if we take it also as an analogy of how much can you expand that, I guess another analogy is with creatine. Even over the short term, in a month, where we do not gain muscle except whatever water comes from that creatine, we're able to expand the creatine pool, 30 or 40%? Somewhere around that. And so you can imagine for vitamin B5, we don't know what it is, but look, we can guesstimate that based on my rat data. I guesstimate 4.5 grams. Based on the short-term studies of 100 milligrams a day for a week, 60% of that dose is retained. Based on a 45 milligram injection, 60% of that is retained. Based on the studies that Alex cited in the last episode, or in the last episode in this series, 5.6 grams of megadoses of pantothine might be retained. So we can imagine that our total body storage is grams, and that probably we can improve our store by grams, and without putting more detail on it than that. So if we put some numbers on this, and we imagine that the total body stores of an average person, of an average adult weight, are 4.5 grams, what we lose in the urine would be about 0.1% of our total body stores. Well, no wonder 9 weeks of zero intake only produce fatigue and obvious loss of enthusiasm. No wonder, because what do you lose on a zero B5 diet for 9 weeks? About 6% of your total body stores. And if you're thinking of big doses, 100 milligrams would only be 2% of your total body stores. If your capacity was to increase your total body stores by double or by 30% even, then taking doses far above what you could get from food would be able to be effectively retained for quite some time. Also, think about if it takes this long of a moderate deficiency to drain total body stores. Many of us may have been walking around for years of consuming something where we're just losing 1 milligram a day out of our total body stores. And we have a very long history that has set us up to, at least over the course of weeks or months, benefit from supplementation with much higher doses than we would expect to get from food. So one thing that's difficult to say about any of the studies on how much we should be consuming is that many of us might not be expected to just need maintenance doses from food. We may have had a history of deficits that we need to make up from. With that said, if we look at some of the studies that have been done, one thing that I would want to see is there any indication that something changes about the relationship between your intake and your urinary excretion at some level of intake? For example, is there a level of intake at 10 milligrams a day or 15 milligrams a day where all of a sudden you're excreting the overwhelming proportion of the dose? And in all the studies that have been done looking at that, none of them found any such change over the range of 1 to 10 to 12 milligrams per day of intake. So knowing that at 4 milligrams per day, you're probably losing pantothenic acid every day, and looking at what you could possibly get from food, then I'm inclined to say that we probably want something on the order of 10 to 15 milligrams per day. And if we do get that, two things. Number one, if you do measure your urinary pantothenic acid, that would probably correspond to about 5 milligrams per gram creatinine. And then number two, going forward to looking at how we get pantothenic acid from food, one of the targets that I'll look at is how to get the Institute of Medicine's adequate intake of 5 milligrams a day, and also how to get what I think is a reasonably higher target of 10 milligrams a day. I think that we know a lot less than we would like to know about pantothenic acid requirements, and probably less than for any other B vitamin. But we can say a few things. At the AI of 5 milligrams a day for most adults, 6 milligrams per day for pregnancy, and 7 milligrams per day for lactation, that's probably just under what's consistent with long-term protection against frank deficiency. You probably want at least a little bit more than that. Given the lack of evidence for extrapolations to children, if you're going to favor the AI of 5 milligrams a day for adults or my idea of 10 milligrams per day for adults, I would consider this to be based on a 2,000 calorie diet. So for a child, if you want to decrease the amount, you could say, all right, we want 2.5 milligrams per 1,000 calories following the AI, or we want 5 milligrams per 1,000 calories following my recommendations. Keeping in mind that the only data that we have suggests that children and adolescents, because they're growing, actually need more pantothenic acid, not less. And so it might be the case that you really want to focus on getting as much as you can from food in a child. I also think that because there's such dramatic uncertainty about, number one, what are the maintenance needs? And then number two, what is the potential requirement to correct years of suboptimal intake? Then I think it makes sense to say, look, I don't have $800 to run an organic acids test, and yet I am eating 10 milligrams a day, and yet I have fatigue, I have mood problems, I have insomnia, or I have tingling in my hands and feet, or I have gut problems, or I have cramps. I think if you have signs and symptoms of pantothenic acid deficiency, giving the complete lack of evidence for any toxicity syndrome, then I think it does make sense to use high doses to see if they help. And those doses could be hundreds of milligrams per day. I always think that you should start low and work your way up. So I think it's better to start with something on the order of 10 milligrams per day, or to go to 100 milligrams per day before you go to a gram per day. But I do think it's safe to experiment with higher doses. So how would we get any of these targets from diet? First, we have to consider some general considerations for diet. So the bioavailability of pantothenate from food is probably 50%. And that's based on canned foods that were homogenized into a goop. But in the study that made that estimation, they did analyze the B5 contents after the food was canned and homogenized into a goop. And so the results are probably pretty accurate. Now, there seems to be pretty dramatic losses of B5 with various types of processing in foods. There was a study in 1971 that was not a controlled study, and we can't place high confidence in the details of the numbers, but they took over 700 foods that were published in the USDA database, and they compared the fresh foods to the frozen foods and canned foods. And they found that for fish and shellfish, the frozen foods had 20% less than the fresh foods. For canning, there was 20% less as well. For meat and poultry, they didn't have freezing numbers, but they had canning numbers, and there was 26% less in a canned meat. For dairy products, there was 35% less in the canned milk. For corn, there was 50% less when it was canned and 19% less when it was frozen. For root vegetables, there was 46% less when it was canned. For potatoes, 37% less in frozen. For legumes, 78% less in canned. 78%. 57% less in frozen. Very strong argument for dried legumes here. For vegetables, 56% less in canned, 48% less in frozen. For fruit and fruit juice, 51% less in canned, 7.2% less in frozen. In refining of grains, the refining process removes half the pantothenate, but pantothenate is not one of the nutrients that is added to enriched flour. So refined grains, even if they are enriched, are a major risk factor for having low pantothenate status. Now, this paper was not a controlled study, and we don't know to what degree it's canning and freezing versus other factors in the production of those foods, but there are some controlled studies. They show that freezing is not that important as freezing itself. So in breast milk, over long-term storage, there's 14% loss of B5 over 3 months when it's slow frozen, and there is very good stability over time when it's quick frozen. So quick freezing is better. But the thing with frozen vegetables is it's the blanching loss that kills them. When vegetables are frozen, they are blanched first, and they can lose up to 50% of the B5 during blanching. For example, soybeans, when they are blanched, lose 50% of their B5. In spinach, they lose 64% in water blanching, but only 13% in steam blanching. Broccoli loses 62% in water blanching and 45% in steam blanching. Once the vegetables are frozen, long-term storage, they lose a smaller amount. So the blanched broccoli, for example, it lost 62%, and then it lost another 24% when it was under 60 days of frozen storage. After that, the loss leveled off. Now, it depends on the food, though. So, for example, in sweet potatoes, boiling them didn't destroy any B5, but baking them at a higher temperature destroyed 23%. Whereas in peanuts, boiling them for 40 minutes caused a 25% loss. Roasting them at 180 degrees Celsius destroyed all of the B5, but roasting them at 160 degrees Celsius only destroyed 25%. So rather than trying to memorize all these numbers, and actually I should point out one more thing. For fava beans, sprouting them, even though it increases some nutrients dramatically, it reduces the B5 by 19%. So rather than memorizing all these numbers or trying to make rules out of it, the rule that we derive from this is that because the effective processing is so incredibly strong, when we're looking at databases, we absolutely cannot use data for raw foods and assume that there will be a similar amount of B5 in the cooked food. We have to look at data where it's actually been measured from the cooked food the way that we are cooking it. So with that said, using only foods that have been cooked the way people usually consume them, I analyzed the data from the USDA database and came up with 5 tiers of how to get B5 from food. Tier 1 yields 10 milligrams in 1 serving. The only food in Tier 1 is 2 heaping teaspoons of unfortified nutritional yeast. Now, fortified nutritional yeast I'm not including here because I'm just looking at natural foods. In Tier 2, these are foods that yield 10 milligrams in 2 servings. Here we have the liver of chicken, beef, lamb, or veal, and we have sunflower seeds. That's it. In Tier 3, these are foods where 3 servings would yield 10 milligrams. Here we have beef pancreas and beef kidney, G-toast cheese, I don't know if I'm pronouncing that right, black and red caviar, the kidney from lamb imported from New Zealand, not the kidney from other lamb, pork liver and most products made from pork liver, shiitake mushrooms, and canned grape leaves. In Tier 4, we jump down a lot. We give up on trying to get 10 milligrams, and we say, okay, I want to get the AI, 5 milligrams a day, recommended by the Institute of Medicine Food and Nutrition Board, in 5 servings per day. On this list, we have some things that a lot of people eat. We have canned chili, peanuts, peanut butter, cashews, mushroom, in the case of white mushrooms and portobello mushrooms, raw avocado, salmon, trout, if they're fresh, eggs, And then we start moving into things that most people don't eat. We have duck and goose. We have pate. We have beef, thymus, and heart, pork, kidney, and brain, emu meat, and the giblets and heart of chickens and turkeys. Many cuts of steak and other fresh meats do go into tier 4, but many don't, and the data is very messy. Tier 5 is you have a good chance of hitting the AI of 5 milligrams if you eat 4 pounds of these foods in total. In other words, if you eat 4 pounds of food a day, like the average American, and all of it is these foods, you will hit the AI. This food has most cuts of fresh meat that are not in tier 4. It has whole grains, most natural dairy products besides butter, most seafood that is not canned, most beans that are not canned, roasted almonds, raw coconut, most other nuts and seeds, but not all, most processed meats, most spices, and many vegetables are in tier 5, but many are not, and the data is very messy. The foods that you know are going to make you not hit the AI for B5 if you don't eat any of the foods in the top 3 tiers, which is basically liver, some organs, and nutritional yeast. If you eat any of these foods and you don't eat the superfoods, you will not meet the AI. We have sugar. We have fat, added fat, refined flour, many canned goods, many heavily cooked goods, and heavily processed foods. Right? These are the foods where if we do not eat the superfoods and we do not take a supplement, we do not take a multivitamin, and we eat a meaningful amount of those foods, sugar, fat, refined flour, canned, heavily cooked, heavily processed foods, you will probably not meet the AI for vitamin B5. Now we come to the question of the intestinal bacteria. This is the first time where I really questioned whether the intestinal bacteria may be a major source of vitamin B5. And I still don't know the answer. But I think compared to every other B vitamin that I've looked into, this is the one where the case is most compelling. So let's review the case. In 2016, Gomenak published a paper that laid out the case for the microbiome being a major supplier of Pantothenic. In his case, he was using high-dose vitamin D to fix sleeping problems in a case of 1,000 neurology patients. And their sleep problems came back after a while of being on the vitamin D supplement. He speculated that the D may have disturbed their microbiome, and he added a B complex. And this made the sleep better. And so he, looking at his data, this is a hypothesis paper in medical hypotheses. He doesn't have a strong case from his data, but he hypothesized that maybe what was happening was the microbiome that should be making B vitamins needed, because it was killed off in these patients, they needed extra B vitamins to jumpstart their intestinal microbiome. So I think from his data, the case is weak, but he cites some interesting evidence. So his claim is that in modern life, where we're living indoor lifestyles, we have a pandemic of vitamin D deficiency and the wrong microbiome. And he argues that maybe gut microbes are needed to help us digest coenzyme A in foods, that gut microbes might require vitamin B5, such as lactobacillus, and may steal B5 from us if we're engaging in probiotic supplementation unwisely, or that microbial synthesis is supplying us with B5. And when I read this paper, I'm not too convinced, because I think his data are very compatible with just vitamin D and B vitamins help sleep, and that's it. But I do think it's interesting that gut microbes could steal B5 and they could make B5. I also think that the hypothesis is interesting that you may need exogenous B vitamins to affect your intestinal ecology in a way that promotes B vitamin synthesis. And I'll come back to that after I talk about one more paper. So Sibin and Strauss in 2016 made a stronger case for this. They pointed out that the lactobulgaricus factor that was known about in the early 20th century turned out to be pantothine. This was a factor that Lactobacillus bulgaricus was producing in large amounts to feed other lactic acid bacteria. And so the form of supplementation that Alex was talking about, pantothenic, is produced by Lactobulgaricus in very large amounts, and it is used to supply B5 to other lactic acid bacteria that cannot make it themselves. And so this is an example of where if your B vitamin producing microbiome gets killed off, then you might need to supply those B vitamins to get those other lactic acid bacteria actually produced. Because you have an ecological network where even though in net they are B5 producers for you, there are so many members of that network that depend on this central hub in Lactobacillus bulgaricus, where if it's not there, the whole ecology suffers and your net B vitamin production just gets destroyed in your gut because of the lack of that ecology. There are other microbes that might produce pantothenic, such as E. coli. A number of human pathogens and symbiotes cannot make their own B5 and may be net stealers of B5. The bacterium that causes syphilis may be a net stealer of B5. Gardnerella vaginalis, which causes bacterial yeast infections, may steal B5. Some mycoplasma and bifidobacteria and chlamydia steal B5. And bifidobacteria are considered good bacteria. But if you don't have Lactobacillus bulgaricus in your gut and you don't have a lactobacillus community, which in fact I found out through uBiome is exactly what my gut looks like, and you have bifidobacteria, which steal B5, which I have 3 times more than the average person, my gut bacteria looks like it's a net stealer of B5 because I have so much bifidobacteria and I don't have a lactobacillus community. Now, the one study that seemed compelling at first, that Sibin and Strauss cite, is that mice on B5 deficient diets only develop severe problems when given antibiotics. Now, one of the things you have to keep in mind whenever you're trying to generalize from mice and rats about the microbiome is that mice and rats eat their own feces. So if you go back to this paper, they say that they put the mice in a wire mesh cage so that the feces would drop between the wire meshing and the mice would eat less of their own feces. They don't say they did that so they wouldn't eat their own feces. They say they did that so they'd eat less. And when you look at why they gave the antibiotic, the reason that they gave is that they knew the animals were eating their own feces, and so they wanted to eliminate the vitamin B5 that would be produced in the feces that the animals would eat. So I looked up, how do mice eat their own feces? Well, there are videotaped examples of how each species eats its own feces. So for example, rats will poop into their hand, they will bend around and poop right into their mouth to eat their feces. In mice, they can't reach with their mouth, but they put their forelimbs, they're like our hands, right? They put their forelimbs right up to their anus and they take the feces, then they bring the feces back to their mouth and they eat it. So you put them in wire mesh cages and they can't eat the feces that falls between the wire meshing, but they can eat they eat 50 to 60% of their feces right by taking it from their anus. And then on top of that, they will eat the feces that sticks to the wire meshing. And when you put mice on B vitamin deficient diets, this was studied in the case of B12, put them on a B12 deficient diet, they double how much feces they eat. So I'm sure they do when you put them on a B5 deficient diet. In fact, the only way to prevent a mouse from eating its own feces is to make them wear a plastic tube that prevents them from bending their torso so they can't reach around to their anus. And even then, they will still eat the feces that falls in the wire mesh caging. So you have to make the wire meshing very big so that very little falls on it. So if we come back to the mouse study, it's not convincing that they developed B5 deficiency only when given antibiotics. But there are some elements of the evidence that are very compelling. So number one, gut bacteria can make pantothenic acid, and some of them secrete it for use by other bacteria. If they were just making it for their own use, maybe you wouldn't absorb it at all. But if Lactobacillus bulgaricus makes it 4 other bacteria, then it's freeing the B5 into the environment, and it should be available for absorption. Gut bacteria also may scavenge B5 and steal it from you. So if both those things are true, it makes sense that your gut microbiome is going to affect your B5 status. I would add other evidence to this. Cells in the colon do possess a sodium-dependent transporter that transports vitamin B5. Now, some other things that made me think that the microbiome might be important is that in the deficiency cases, they had to use an antagonist to produce severe deficiency in an experimentally controlled setting. And so I thought, why is this? Well, maybe what that antagonist is doing is preventing them from activating B5 that's produced by their microbiome. Why is it so hard to produce a deficiency with just dietary restriction alone? And then there are these studies that used very high doses that Alex will talk about to benefit certain things. Why would you need high doses that are 100 milligrams to actual gram amounts? Why would you need those to have benefits if food doses are 5 to 10 milligrams, unless the microbiome is a major producer? So these things did make me think, but most of them are not that convincing. So you could easily explain the difficulty of inducing a deficiency over the short period of time when you have such massive body stores. That alone can explain almost all of those things. And if we know that we could have years of deficiency and high doses can make up for that, then even doses of gram amounts might be correcting years and years and years of suboptimal status. Still, the convincing parts are gut bacteria can make B5, gut bacteria can steal B5, and we do have B5 transporters in the colon. Still, there's a very powerful argument to me against the microbiome being a major source in basically everyone that's ever been studied in the last 100 years. And that is that normal urine concentrations are around 3 to 4 milligrams per day. And they drop to 0.8 in response to 9 weeks of consuming no B5 at all. They almost double in response to 10 milligrams. If the microbiome is the major supplier of B5, why is it that the urinary concentration drops 80% over the course of weeks of no dietary B5? And why is it the case that something like 5 or 10 milligrams makes such a huge difference in your urinary concentrations? This indicates to me that in the last 100 years of people who have been studied, that the microbiome makes maybe a minor contribution to B5 status on the order of 10 to 20%. Now, it's possible that if everyone over the last 100 years has had the wrong microbiome because of modern lifestyles and industrialization, that there could be a quote-unquote lost microbiome of ancestral days that none of us have, that we should have, and that our bodies are designed to live on the high amounts of B5 produced by this lost microbiome. But there's no evidence of that, and that's very speculative. So what I think is, yes, the microbiome probably contributes 10 to 20% of a person's B5 status. A bad microbiome may make things a lot worse by not giving you that 10 or 20% and by stealing B5 from you. So the microbiome is relevant. But I think it's very, very, very hypothetical to suggest that maybe we all just have the wrong microbiome, and in some faraway microbiome of times past, that the B5 produced in the gut was the major B5. If that's true, it has not been the major B5 over the last 100 years. So what might be causes of suboptimal status or deficiency? Well, remember, we don't know that much because our working assumption of the last 100 years has been that no one develops natural deficiency, and so it's very under-researched. But some things that we do know. Ethanol is an inhibitor of pantothenic kinase. It seems that in alcoholics, they have adaptations that prevent them from having serious problems, but ethanol does act as the antagonist used in the 1950s to produce clinical deficiency, or as the genetic defect that I discussed in the last episode in the series pantothenic kinase deficiency, ethanol does mimic that to some degree. There's an old paper showing that thiamine and riboflavin increased pantothenate levels in the urine. There were no statistics in this paper. I don't know how much we can trust it. Probably those people had very mediocre status of all these nutrients based on the urine concentrations. But that gives proof of principle that you might have your pantothenate status hurt by deficiencies of other B vitamins. That makes sense in a couple ways. One is that all the steps in activating pantothenic acid or most of them are dependent on ATP. You need the other B vitamins to make your ATP. This comes back to what we talked about in niacin and riboflavin, where we said things that hurt your ATP status, like hypothyroidism, insulin problems, undereating, use of metformin or berberine, these could theoretically compromise your pantothenate status because you can't activate it as effectively. The math suggests that high-fat diets would slightly increase B5 needs because you use 20% more B5 when you burn fat compared to carbs. The mouse study that I cited in the last episode in this series, where in a model of the genetic defect in pantothenate kinase, they had very slight neurodegeneration on a normal diet, but devastating neurodegeneration on a ketogenic diet, suggests that if you have very bad B5 status or you have an impairment in B5 metabolism, that perhaps a high-fat diet would be more harmful than that 20% math should suggest. Finally, there are many inborn errors of metabolism that cause backups in metabolism that hog the CoA pool. These don't cause B5 deficiency, but they effectively cause a functional B5 deficiency because they deplete you of CoA available for the reactions that you need it for. And most commonly, these are disorders of fatty acid metabolism or amino acid metabolism. What happens is, let's say you're burning a fatty acid, you burn that fatty acid part way, you've attached the fatty acid to CoA to make a fatty acyl CoA, you've started chopping off acetyl CoA from it to enter into the rest of metabolism, and you're stuck with these fatty acyl CoAs where you hit a metabolic block and you can't go any further. The fatty acyl CoAs accumulate, and now there's no free CoA left to continue those reactions. And that causes all sorts of effects that would be very much like the effects of vitamin B5 deficiency in a very severe way, because you've depleted yourself of the ability to use coenzyme A. Most of those disorders are helped a lot by avoiding fasting and feeding high carbohydrate diets. And that's because you use less CoA. There's less stress on the CoA pool. And you have more roundabout ways of getting your energy when you use carbs than when you use fat. In addition to genetic defects, all of which incidentally are very rare individually, but are substantial collectively. Apart from the genetic defects, deficiencies of biotin will elevate levels of propionyl CoA, which is derived from the amino acids methionine, threonine, or the branched chain amino acids. If your propionyl CoA goes up, your CoA pool gets hogged because it's all sequestered as propionyl CoA. If you are B12 deficient, you will elevate methylmalonyl CoA, which is derived from propionyl CoA. And if methylmalonyl CoA elevates, your CoA pool gets hogged as methylmalonyl CoA, and you become deficient in the CoA needed for all the other reactions. If you are riboflavin deficient, then the amino acids lysine and tryptophan can lead to elevations of glutarol CoA because they can't be fully broken down. And because riboflavin is so important for fatty acid oxidation, you can have fatty acyl CoAs that elevate because they can't be broken down. Those acyl CoAs accumulate, and you're left without free CoA to engage in the metabolic reactions that you need. And so even though these disorders and severe nutrient deficiencies are not super common, they will at least be rare cases of functional B5 deficiency. But also, my suspicion is that a lot of people may have some cap on their metabolic pathway. Like, you may only be so good at metabolizing branched chain amino acids, but because you're eating a very high protein diet or you're downing tons and tons of whey protein, for some people, you may surpass your metabolic capacity in a way that causes a very moderate problem of this nature and effectively induces a vitamin B5 deficiency, a functional vitamin B5 deficiency. Unfortunately, nothing can really be said about the prevalence of suboptimal status, because in all these studies that we're looking at, like when I talk about they looked at dietary intake versus urinary excretion, none of those studies reported surveys of fatigue or loss of enthusiasm or gut problems or cramping or any of the signs of vitamin B5 deficiency. So I think it's a very open question of whether there exists natural suboptimal status, and my suspicion is that there does. So with that said, I'll conclude my contribution to this by saying I think supplementation with high doses is warranted, even if you don't have data, and even if your diet looks fine. If you have any of these signs and symptoms, fatigue, feeling down in the dumps, low energy, insomnia, grumpy mood, cramps, gut problems, if you any of those things, I think, are reasons to say, you know what, maybe I should try some higher dose B5 and see if it helps. And so with that, on the topic of supplementation, I'll bring it over to Alex. Alex, what can you tell us about some of the benefits besides fixing deficiency? What are some of the benefits that we might get from supplementing with pantothenic acid or its derivatives?
Yeah, so when I was looking around at what pantothenic acid is used for supplementation, there isn't a lot out there. And what is available is heavily concentrated in two areas. The first being as a therapeutic for elevated blood lipids. And this is specific towards the form of pantothenic acid called pantothine, which is basically two pantothene molecules bonded together by a chemical bond between two sulfur molecules. And this does contribute bioavailable pantothenic acid to the body, but pantothenic acid itself doesn't appear to have the same effects. So what caught my attention with this, first and foremost, was a review article in 2005 that looked at 28 clinical trials in patients with dyslipidemia. And it reported that using pantothine supplements reduced LDL cholesterol content by 10 to 20%. And reduced triglycerides by 14 to 33%, and increased HDL cholesterol by 6 to 8%. And it was an interesting review because the studies that it included were all published within an 11-year period, beginning in 1981 and going through 1992. And there wasn't any clinical trials published in the 12 years up to the publication of this 2005 review article. And of these 28 studies, 25 of them were conducted in Italy, which is again strange because pantothine was first approved for lowering blood lipids in Japan, not Italy. But only 3 of the trials were in Japan, and the other 25 were in Italy. So overall, the studies lasted 3 to 48 weeks with an average duration of 12 weeks, and they used 600 to 1,200 milligrams of pantothine per day. And the most common dosage was 300 milligrams taken 3 times per day with meals. And there weren't any severe adverse reactions. As we discussed in part 1, the adverse effect rate was about 1.4%, with the most commonly reported reactions simply being mild heartburn, itchiness, and some diarrhea. And the effects don't occur with pantothenic acid because when the researchers started to investigate the mechanisms of this, it was found that the effects are likely mediated by cysteamine, which is the molecule that is binding the pantothenic acid molecules together. So the way pantothine works is going to be similar to a statin in that it ultimately inhibits HMG-CoA reductase. However, the actual point of inhibition of pantothenic is not at that location in cholesterol synthesis. It's further downstream, somewhere within the 19 steps of cholesterol synthesis between lanosterol and cholesterol. And what happens is the inhibition leads to a accumulation of oxysterols that then inhibit HMG-CoA reductase as a negative feedback loop. And pantothenic also appears to inhibit acetyl-CoA carboxylase, which is used to provide the malonyl-CoA substrate for the biosynthesis of fatty acids, which decreases fatty acid synthesis and can help explain the triglyceride lowering effect in addition to the cholesterol lowering effect. And I mentioned that it was similar to statins, but there is one key difference in that when we look at the adverse effects of pantothenic administration, we don't see changes in serum levels of coenzyme Q10 like we do in statins, which are known to deplete the coenzyme Q10 pool in individuals and therefore requires supplementation to help offset the adverse effects of that. And so the mechanisms through which pantothenic work are similar, but they don't appear to be of the same magnitude as a statin. And the off-stream adverse effects don't occur. There are, no indication that it increases the risk of diabetes or muscle pain or other things that you might see with a statin. Now, of course, the effect isn't as large, but it's also not negligible. So again, in people who have elevated blood lipids, it suggests that over a 4-month period, using an average of 900 milligrams, 300 milligrams 3 times per day, you will see reductions in LDL cholesterol that average 20%, average increases in HDL cholesterol of 8%, and average reductions in triglycerides of 33%. Now, since all of these studies were published throughout the 80s, there have been 2 more randomized controlled trials in North America, the first being published in 2011, that basically saw all of these studies and asked, why don't we have any studies in North America looking at this? And so the researchers wanted to see what the effect was. The difference being that these people didn't have dyslipidemia. They weren't at risk for cardiovascular disease. They were middle-aged adults that were considered to be at a very low risk of cardiovascular disease. In that, for example, their average LDL cholesterol when they started this thing was around 100 milligrams per deciliter. And even in these people, supplementing with 900 milligrams of pantothenic per day lowered their LDL cholesterol by 4%, their ApoB containing particle counts by 5%, and their triglycerides by 12%. Keeping in mind that in absolute terms, these weren't very large reductions, right? 4% corresponds to only 4 milligrams per deciliter. But it's still notable considering that their levels were already considered optimal by current cholesterol treatment guidelines. And in the study, again, they observed no effects on blood pressure, no effects on inflammation, liver enzymes, or coenzyme Q10 values. And there was no difference from the placebo group for adverse events. It was basically small benefit towards blood lipids without any known bad effects. And so in a follow-up study by the same research lab, 3 years later, they did take people who were at risk for cardiovascular disease, and these were people who were candidates for statin therapy. Their LDL cholesterol was around 170 to 180 when they started the intervention, and it was the exact same intervention over 12 weeks. But they saw more pronounced reductions. They saw an average 11% reduction in LDL cholesterol and 8% reduction in ApoB containing particles in the blood, which corresponds to 20 and 10 milligrams per deciliter, respectively. And again, no other effects on blood chemistry other, in this case, minor reductions in liver enzymes and CRP, a marker of inflammation, with no adverse events reported. And so while the studies are largely constricted to Italy, with a handful in Japan, and then now we have 2 studies in North America, overall, they suggest that pantothene is a dietary source of pantothenic acid that is effective at reducing LDL cholesterol and ApoB by about 4% in people with already low levels of blood lipids, and 10 to 20% in people who have dyslipidemia. It also seems to reduce triglycerides and increase HDL cholesterol exclusively in people with dyslipidemia. And it accomplishes these tasks by inhibiting cholesterol synthesis in the liver and fatty acid synthesis with minimal side effects or adverse effects.
So, Alex, having researched this and niacin, what would be your B vitamin of choice to use as a drug to lower lipids?
I think this one. I think that niacin would have a more pronounced effect, but niacin also carries with it the higher risk profile. We don't know of any risks with this. And then as we talked in the other episode about the toxicology of pantothenic acid supplementation, it's basically non-existent. And so, especially at these doses, which were well below some of the doses that were being tested, upwards of 30 grams per day, and here we're talking less than 1 gram per day.
Yeah, to give some perspective here, the required amount of niacin is on the order of 15, 16 milligrams. And the lowest dose used in case reports of liver failure, or modestly, like usually it's 3 to 9 grams a day, but it's been 300 milligrams, as low as 300 milligrams a day has been the lowest case report. Whereas pantothenic acid, the adequate intake is 5 milligrams. I'm recommending 10, but we have studies going up to like 15 to 30 grams of it. So the ratio of zero effect at those extraordinary doses compared to how much is the minimal requirement, like the window between the minimal requirement and the dose that does cause harm in niacin versus the dose that has no evidence of harm in pantothenic acid, it just seems like a pantothenic acid is just dramatically more safe, it seems, at high doses.
Yeah, definitely. And it has an effect in people who are already healthy for blood lipids. So it's really kind of seems to be a default choice for anyone who just wants to ensure optimal cardiovascular health. There doesn't seem to be a downside to doing it, at least not one that we know of. So with that, the other most widely researched use of pantothenic acid is for acne and general skin health and wound healing. And these studies use primarily topical application, except in the case of acne. So with regards to skin health and wound healing, there is a compound called dexpanthenol, which is basically an alcoholic analog of pantothenic acid that was first introduced as a skin ointment in 1944, called bepanthen. And today you can find a variety of dexpanthenol preparations available: creams, emollients, gels, lotions, oils, and so forth. But when applied topically, dexpanthenol is readily absorbed in the skin and converted to pantothenic acid, which is then available to produce CoA. And this is important because the skin depends on a mixture of sphingolipids, cholesterol, and fatty acids to maintain its integrity and that water barrier. And in particular, the creation of sphingolipids depends highly on the concentration of CoA being available to condense palmitoyl-CoA with serine. And so with this, it provides the kind of basis for why this was investigated in the first place, is because this cream is absorbed very effectively into the skin and it provides the necessary substrate to maintain skin barrier integrity. And so we do see that in clinical trials looking at its effects on overall skin health. We know that dexpanthenol acts like a moisturizer with skin barrier improving properties, and it may go above and beyond that to also facilitate wound healing. So there have been studies showing that its application increases the expression of genes that are involved in skin proliferation, inflammation, and tissue repair. And it's been shown when you apply it to like rashes and the such on the face that it's beneficial in reducing inflammation. Both that type that's caused by a like toxic external chemical that causes an irritation on the skin, and also signs or types of inflammation that come about endogenously from like a nutrient excess or deficiency, such as cheilitis, which is inflammation around the lips. And even when we apply, when we apply it on the inside of the mouth as well, using a lozenge, where we take the lozenge, we suck on it, we let that dexpanthenol kind of coat the inside of the mouth and the throat, we see that it accelerates mucosal healing and reduces throat pain after tonsillectomy in children. And it can also help facilitate the healing after tattoos. So the studies we have are scattered and they're kind of random, frankly, right? You go from tonsillectomy to tattoo removal to chemical-induced like burns, but they all point in a consistent direction that topical application of dexpanthenol facilitates the healing process and reduces pain and sensitivity in the area, which might be secondary just to its effectiveness as a moisturizer. Now, the other thing I mentioned was acne, and it's been hypothesized that pantothenic acid deficiency is related to acne. And the rationale is basically that puberty results in a greater demand for CoA, and any shortage of dietary pantothenic acid would result in a less than efficient rate of fatty acid metabolism that would give rise to the deposition of fat droplets in the sebaceous glands of your hair follicle ducts, which would result in acne. And so we have a handful of studies. For example, the one that came up with the hypothesis to begin with, after generating that hypothesis, they took 100 Chinese kids and they reported near immediate, and I mean within 1 to 2 days, reductions in sebum secretion on the face with reduced acne and a regression of lesions within 1 to 2 weeks. And complete control of acne within 8 weeks using 10 grams per day of oral pantothenic acid taken in 4 divided doses, plus using the dexpanthenol cream, applying it to their face 4 to 6 times per day. And over an 18-month follow-up period, they found that the acne remained under control as long as the students maintained a dose of 1 to 5 grams of pantothenic acid every day, with higher doses required for those who started with more severe acne. And so this study got followed up with 2 other trials, and one was first a pilot trial in people with mild to moderate acne that did 8 weeks of supplementing with something called Pantothenic, which provided 2.2 grams of pantothenic acid along with other B vitamins and a small amount of L-carnitine. And they found that in this small group of people, after 8 weeks of supplementation, the average number of skin lesions present on their face were reduced from 20 down to 11, which came with subjective improvements in quality of life. So these researchers conducted a larger study to follow up on that one over a 12-week period. And in in this study, they recruited people with moderate acne that had more than 50 non-inflammatory lesions and up to 50 inflammatory ones. And they reported using that same pathogen supplement regime that overall skin lesions were reduced by 68%. So again, minimal studies here, but they all point in a consistent direction where pantothenic acid supplementation appears to benefit individuals with acne when supplemented in relatively high doses of 2 to 10 grams per day. And a topical application of dexpanthenol cream seems to benefit skin health and may accelerate wound healing. And the dexpanthenol creams, for those of you interested, were about 5% dexpanthenol. So a couple other areas where pantothenic acid has been investigated, but where things have not panned out, would be in exercise performance. So pantothenic acid has been investigated for its role in sports performance, since it's required for the production of coenzyme A, which is required, among other things, to transport fatty acyls into the mitochondria so that fats can be used for energy production. And it's been proposed that a reduced availability of cytosolic free CoA could be a rate-limiting step for fat oxidation during exercise. And we only have 3 studies using 1 to 1.8 grams per day of pantothenic acid for 1 to 2 weeks, and all of them failed to note performance improvements in a cohort of recreationally active men, a cohort of trained cyclists, and a cohort of trained distance runners, using tests such as a 50-kilometer submaximal cycling, a 2,000-meter time trial, and cycling to exhaustion at 75% of VO2 max, plus power output. There were also no changes in blood chemistry, including respiratory exchange ratios, which would give an indication of how much they're relying on fatty acids. And there were no changes in perceived exertion or free CoA levels in skeletal muscle. One of the studies actually took muscle biopsies, and there was no change in free CoA levels within the skeletal muscle. So what limited data we do have suggests that supplementation with pantothenic acid will not benefit endurance exercise performance.
This seems fairly analogous to riboflavin, right? So like riboflavin, the evidence was pretty much indicated that there wasn't really an effect on performance. But yet, where we had a very good marker of riboflavin status, and we had very good cutoffs that were well established from a lot of research in the field, we were able to see things like when you do cardio 6 days a week, your riboflavin requirement goes up 60%. And when you lose weight, it goes up 60%. When you do both, it doubles, and things like that. And so it seems like in this case, we have this century of baggage of believing that natural pantothenic acid deficiency doesn't exist. So we have no idea how to go about saying, well, you may not need this for performance, but did you performing increase your requirement in other ways? For example, like I don't know if this is plausible or if there is data, but maybe in some subset of the population, the teenagers get acne more when they're involved in certain sports because look, your body is your body's not going to poison itself by limiting the supply of CoA to be able to handle the influx of fatty acids. That's what happens in fatty acid oxidation disorders, and that's why people that have that often don't survive bouts of fasting and exercise. You're like the limit, having a limitation of the CoA pool is poisonous to your metabolism for reasons that I won't go into now and explained in episode 1 of this 2-part series. But it makes total sense that your body would, and also there are studies where the heart, for example, and I believe this is probably true in skeletal muscle, has a very large reserve of pantothenic acid that's just sitting there waiting to be turned into CoA. And the heart can, boom, like make that CoA real fast if it needs it. And so I think that what we're seeing here is that when you engage in exercise, your body is going to prioritize fulfilling the requirements of that exercise, and it's probably coming at the expense of something. But because we have so much less research here on the requirement than riboflavin, we just don't have the tools we need to figure out what that is.
Yeah, definitely. And it is certainly possible that exercise would increase requirements, not just from an energy metabolism standpoint, but simply because a secondary effect of exercise would be more muscle tissue. And if you have more muscle, then you're going to need more CoA to maintain it. But something else to keep in mind as well with the very, very limited pool of studies we have on exercise is that every single performance outcome they were testing was primarily glycotic, right? When you're doing a 2,000 meter time trial or cycling to exhaustion at 75% of VO2 max, or even competing, like actually competing for a 50-kilometer cycling, you're going to be relying primarily on glucose as an energy source. That is the only energy source, notwithstanding ATP and creatine phosphate. But that's the only energy source that can maintain high levels of exercise intensity. And so if the entire hypothesis...
So I think that your level of intensity is going to be super dependent on glucose, but you are still going to burn a ton of extra fatty acids in a marathon, right?
Yes, 100%. You're always going to be burning some fat, but the point is that your body will preferentially, the more that you push yourself, rely on glucose. And in fact, it's a reliance on glucose that lets you push up to higher intensities. If you didn't have any glucose available, then your perceived high intensity effort would not be as actual high intensity as if you did have glucose available.
Yeah, and I think this kind of relates to something a point that I made at the beginning of part one in here, which was when the B5 pool is limiting, if you have enough carbohydrate, you can just run anaerobic glycolysis and say, I don't care about my CoA levels. But if you don't have the carbohydrate, then you can be in trouble. And so in the mouse study that I cited twice in the course of this two-part series, it was where they had a genetic defect in pantothenic kinase, and their neurodegeneration was not that bad on a regular diet. And then they went on a keto diet, and all of a sudden it was devastating. And so, it's because even though it's because when you are put under conditions where you are under a lot of stress, if you have carbohydrate where you can derive energy quickly without using CoA at all, the stress of having a limited CoA pool just isn't that stressful. It's, what would happen if they repeated those studies on a ketogenic diet? Then the B5 may have proved to be limiting.
Yeah, and so the whole point I brought up the high-intensity glucose thing, it was simply that the entire hypothesis of pantothenic acid supplementation was to improve fatty acid oxidation during exercise. But all of their tests weren't stressing the fatty acid pool. They were all using glucose. And so it is possible that supplementation could benefit performance at lower intensities or in people doing events like ultra marathons. But we simply don't have that data available. And so we can't do much more than, logically speculate. So another area, if we come back to kind of the aesthetic realm with skin, another area is hair. And pantothenic acid deficiency in rats was shown to cause graying of the hair since at least 1946. And we don't have any human data available, but other than the fact that the elimination of pantothenic acid in the urine doesn't seem to differ between people with normal hair and people who have graying hair or hair loss. The only relatively modern evidence for pantothenic acid on hair health involves one study that measured hair variables after they put a product in their hair and let it sit there and dry, which, is difficult because it's neither really an oral supplement nor a form of shampoo. It's like you get out of the shower and then you put this product on your hair and then you just let it sit there for the rest of the day. And the product contained pantothenic acid along with some other nutrients, and it showed that hair fiber diameter increased along with its flexibility and break stress. But we can't really draw any conclusions about the role of pantothenic acid because it also had things like caffeine, nicotinamide, dimethicone, and an acrylamide polymer. So it's like, okay, maybe, but we can't form any conclusions there. And finally, the last thing that's been investigated for is arthritis. And the first report I could find was published in 1953, titled "The Therapeutic Advantages of the Addition of Calcium Pantothenate to Salicylates in the Oral Treatment of Rheumatoid Arthritis." And the final report I could find was published in 1980, titled "Calcium Pantothenate in Arthritic Conditions, a report from the General Practitioner Research Group." I couldn't locate the full text of either. And since then, this research has pretty much dropped off the map, and there's good reason for that. So an article in The Lancet in 1963 found that arthritic patients had low whole blood levels of total pantothenic acid. That's including free and bound forms. And it didn't matter what type of diet the arthritic patient was on, whether they were vegetarian or omnivore. Compared to omnivores and vegetarians without arthritis, they had lower levels. And within this article, they mention a study where daily injections of 50 milligrams of calcium D pantothenate alleviated symptoms of rheumatoid arthritis after a week of treatment, with no further improvement after 3 weeks. And whole blood levels rose from the levels that they observed in their arthritic patients up to the levels they observed in the omnivores and vegetarians. And the levels fell after they stopped supplementing, and the symptom severity returned to baseline. Now, in response to this article, a physician in a communication published that he had been successfully treating patients with osteoarthritis using 12.5 milligrams of oral pantothenic acid in the morning and evening for 25 milligrams per day. And that the benefits usually took 1 to 2 weeks to manifest, but disappear when they stop supplementing. And he mentions studies in rats that gave him this idea that showed joint changes resembling osteoarthritis when they suffered pantothenic acid deficiency. And he suggests to the original article author that, it might not be the rheumatoid component of the arthritis, it might be the osteoarthritic component, since rheumatoid arthritis has components of osteoarthritis in it in terms of joint degeneration and bone-on-bone contact. But it could also just be a placebo effect. And the reason I say that is because somebody finally decided to conduct a double-blind controlled trial in 1971 involving 40 patients in their mid-60s with osteoarthritis of the knee. And they gave one group 25 milligrams of calcium D pantothenate each day and compared it to placebo for 12 weeks. And they assessed symptoms both by a physician as well as the self-reported symptoms of the patients themselves. And there was no difference between groups at any time point. And so the authors...
How do those doses compare to the ones that are effective for acne?
These are much lower doses. So, yes, a limitation that I will get to. But the authors suggest that if low levels of pantothenic acid is confirmed, that the deficiency appears to be a result of the disease rather than causing the disease. But like you just brought up, these are really low doses. Granted, they're like 5 times greater than the current adequate intake, but still really low compared to using 900 milligrams per day of Pantothenic or, 2 to 10 grams per day for the treatment of acne.
So I think one of the huge limitations here that we have in even trying to understand what the dose means is the fact that the total body pool is so large, and that what might be relevant is the repletion of the total body pool. In other words, these high gram doses it's very possible, based on what we both reviewed, that most of those doses are actually effectively repleting the whole body pool. Like at those gram doses, you might have grams that are repleting the whole body pool. And so if you take someone whose whole body pool is depleted 5% from what it should, and you supplement them with 10 milligrams, you may see an effect. But you could take the same exact problem and take a group of people who have been they've been losing 5 milligrams a day of pantothenic acid for the last 20 years. And their pool is like grams under what it should be. And over the course of a short term, your 20 milligram dose seems really high. It seems that's 4 times the AI. And yet, what is it in relation to the total body pool? And that's the thing that I think is like, it's the giant elephant in all these studies, is that your intake is sort of like totally irrelevant almost. It's to what degree is your total body pool compromised, and to what kind of dose do you need to get it back to normal?
Yeah, I agree completely. And I mean, one of the other recurring themes of this 2-part podcast is simply that research on pantothenic acid is very scarce compared to other vitamins in general. And so it is just a kind of a giant gray area. From the supplementation standpoint, what we can say with relative certainty is that taking gram doses is going to restore your body pools pretty quickly. And taking these gram doses is probably going to benefit your blood lipids if you use Pantothenic. Again, it's not seen with regular pantothenic acid. And it could benefit if you suffer from acne. It could benefit your acne. If you apply dexpanthenol to your skin, it can also help with skin repair, wound healing, and hydration, and overall integrity. So I would say that from a supplementation standpoint, those are kind of the big 3. Pantothenic for blood lipids, pantothenic acid for acne, and dexpanthenol for topical wound healing.
Cool. So I'll add a couple things onto that. So I made the case that for some people, and these are navigation around very, very gray areas, but for some people, the food sources of coenzyme A and 4-phosphopantothenic are probably the best sources. That would apply if there's any limitation in your energy status, because you don't even have to have a defective pantothenic kinase. You just have to have low ATP levels or low magnesium levels to make it hard for you to activate that. And maybe there are genetic polymorphisms that we don't know about that mimic the genetic mutations that cause severe diseases that we do know about, that cause, moderate impairments in the ability to get that. If you compare that to what's in supplements, Pantothenic is going to have some of the properties of being more easily converted to CoA, but not like coenzyme A and 4-phosphopantothenic from food would. Pantothenic may be an important part of the gut microbiome production. If Lactobacillus bulgaricus is a major producer of B5 in the humans that have high lactobacilli in their intestines, then there's probably a lot of humans getting a significant amount of Pantothenic. So that's interesting from that perspective. I think on the horizon, we're going to see the development of acetylated 4-phosphopantothenic produced for the genetic defects in pantothenic kinase. I do not know whether that's going to be available as a supplement like acetylglutathione and acetylcysteine, or if that's going to be patented and distributed as a drug at extremely high prices because of the disease treatment possibilities. But if in the future, if an acetylated 4-phosphopantothene comes out on the market, that's probably the one way that you could get food-based B5 or something that mimics the benefits of food-based B5 as a high-dose supplement. Because as it stands, you can only get high doses of coenzyme A and phosphopantothene from nutritional yeast and from liver. And as it stands, you would probably overdose on selenium from the yeast and on vitamin A and copper from the liver if you were to try to achieve very high doses of that. So I think that's interesting. The pantothenic acid, it seems like, is there any reason to use pantothenic acid specifically? Or it is the studied form with acne, right? For calcium pantothenic?
Yeah, that's the form used with acne, but the mechanism is related to the actual pantothenic acid. Whereas, for example, with blood lipids, the mechanisms are likely related to the sulfur groups that are attached to the pantothenic acid, which is why you need to use pantothine. So I would probably argue that just using pantothine in general would be a better form. Although the studies with pantothine for adverse effects have only gone up to 1,200 milligrams per day. And so there is a possibility that pushing higher gram doses that you might need for acne control could lead to more complications, especially since its mechanisms of action are similar to that of a statin.
Yeah, I think that's a really good point. So the way that I would view the safety profile is don't use the form at higher doses than it's been used. So pantothine, you could argue, is if you don't have one reason to know one way or the other, it's definitely going to be better for blood lipids, and maybe it's just as good or better for acne, and just acne, right? Yeah. There's no oral dosing effects on wound healing, right? It's dexpanthenol. And dexpanthenol creams are used topically for wound healing and acne together with the oral dosing. If you're going to go to gram amounts, as in the case of the acne doses, you probably want to use calcium pantothenate just because that's what we know it has. Calcium or sodium pantothenate because that's what we know has the high safety.
Yeah, and I mean, you can always combine them too. So if you want to treat acne while also managing blood lipids, you could take, 900 milligrams of pantothine per day, plus another 1 to 2 grams of just regular pantothenic acid.
Yeah, that makes sense. So some rapid fire questions that are sort of standard to supplements. Does it matter if we take divided doses or if we take it all at once?
Well, the dosing schedule in all of these, well, I shouldn't say all, but in the limited studies we have, hasn't been investigated. But they consistently are the most frequent mode of application, at least when it came to blood lipids, which is where we have the most research available, was 3 times per day with meals. Some studies went 2 times per day, for example, 600 milligrams twice a day to get 1,200 milligrams total. But most used 300 milligrams 3 times per day. So I would say 2 to 3 times per day is probably your best bet. For the acne studies, those used it 4 times per day. Keeping in mind, too, that the higher dose you use in a single acute bolus, the more likely you are to get a side effect like diarrhea.
Yeah, so I guess the jury is out because there's no head-to-head studies of different dosing schedules, but what we have uses divided doses. And I think from a physiological standpoint, I think it's less important than for niacin and riboflavin, where metabolic trapping, meaning the ATP level to just keep it in the intestinal cell available for further metabolism, is super important. I think the evidence is very clear that you can absorb massive doses of this, right? But still, there has to be a maximal rate of coenzyme A synthesis per hour, right? And although you can store a lot of free pantothenic acid in your tissues, there's got to be some limit to how much you will store in a given point. So if I had to place my bets on it, I would say that you want to spread it out just to have a more probability that you are never hitting the thresholds where you've saturated your ability to do something with it, in which case that you're obviously going to pee out what you can't do anything with. From what you've seen, does it matter if you take it with food?
Again, we don't know the actual answer here, but most studies do take it with food, and that could also potentially be one reason why the report of side effects are so minimal. It does make sense with, for example, diarrhea, that if you take a gram of pantothenic acid on an empty stomach, you're more likely to have some GI distress than if you take it with a meal.
Yeah, that makes sense. And I think the principle is kind of similar. Like, I don't think it matters as much as for niacin and riboflavin in terms of retaining it with the meal, because everything we looked at has high retention. But apart from the possibility that you'd minimize digestive concerns, it does. If it matters, it's probably better to take it with food from the perspective of retaining it through doing something with it. It probably doesn't matter that much. It probably does matter more at higher doses. But there's no rational basis for doing the opposite. There's no argument for doing it on an empty stomach. And I also think for most people, it's probably going to be more convenient if their supplements are taken with meals, just because you go to the kitchen, that's where the food is, that's where the cabinet is, and so on. I think that pretty much covers the practical applications there. So I want to end with two quotes from papers that I thought were really interesting that I encountered while doing the research for this. So the first one is from a 2016 paper by Sibben and Strauss, where they are talking about how the dogma has been all through the 20th century that there is one way you make coenzyme A. You start with pantothenic acid and you proceed in 5 steps, and you don't ever start from anything but pantothenic acid. And this has been overturned by the need to overturn it in pantothenic kinase deficiency disorders. And so in a paper about the multiple routes of CoA synthesis, he says, now a series of recent articles demonstrate that cells and some microorganisms in fact have several strategies to obtain CoA. In retrospect, such evidence is present in older literature, but the implications were largely overlooked in the absence of our current knowledge of the myriad roles of CoA. I think this is a super important philosophical framework for understanding research. When researchers do something 100 years ago, they may have found 20 things and been able to interpret 2 of them. And so 18 got lost as loose threads. And now, if we go back to that, we may have the tools we need to understand 9 of those 18 that were lost. But if we don't go back to that old research, we will never know about the threads that were lost. And in going back to the old research, in the one of the original papers in the 1950s on inducing vitamin B5 deficiency with an antagonist, there was a wonderful quote about the individuality of requirements. They said, and the foundation for this paragraph is, the background for it is they're talking about how the different people seem to have different manifestations of the disease with different severities. And they said, experiments such as these, long as rigidly confining tests, but short in man's lifespan, impress upon us the wide range of variation, even in subjects chosen to conform to a standard. What Williams has emphasized as biochemical individuality and Medawar as the uniqueness of the individual, we see in the personal quirks and inconsistent biochemical patterns which bedevil the framers of human nutritional experiments and make exasperating the necessarily unsatisfactory solution of the problems of controls. of the problem of controls. We have no doubt that some of the discordant results among our experiments or within a single test run come from ignorance and poor planning. But the wide variations in normal persons can easily frustrate the best experimental design if the requirement for pantothenic acid should vary manyfold in a small group of subjects. I think this was incredibly insightful long before the days of now, where we are just in our infancy of starting to unravel all of the genetic and other variations in our metabolism and our requirements. And I think the first sentence is worth repeating. Experiments such as these, long as rigidly confining tests, but short in man's lifespan. No matter how long our randomized controlled trial is, it impresses us as scientific researchers when it's longer than the longest randomized controlled trial. But it's still so much shorter than man's life. And we can directly apply this to the claim that pantothenic acid, from the Greek pantos, everywhere, everything, that you cannot become naturally deficient in because it's everywhere and in everything. We can directly apply this to the study of Peggy Fry in the 1970s, where people were put on a pantothenic acid deficient diet, 0 pantothenate, for 9 weeks, and all that it produced was complaints of fatigue and an apparent loss of energy and enthusiasm. Nothing more impressive than that. Well, that study was very impressive in the length of keeping people on a rigidly controlled synthetic diet, particularly within the field of vitamin B5, where we don't have longer experiments than that. It's impressive in that way, but it's not very impressive with respect to man's life. 9 weeks is not very long to give someone to become deficient in vitamin B5 when their total body stores might take 6 years to deplete at the level that you lose every day in the urine, and when there are so many compensations that we have to prevent us from ever getting anywhere near that point. Pantothenate is everywhere and in everything, but there are tremendously higher concentrations of pantothenate in nutritional yeast than there are in liver. And there are tremendously higher concentrations in liver than in most other foods. Pantothenate is in everything, but it's not evenly distributed in the food supply. Pantothenate is everywhere, but is fatigue nowhere? Is apathy nowhere? Is discomfort, uneasiness, and pain nowhere? Pantothenic acid is everywhere, but is quarrelsomeness and sullenness and childishness and spending the whole day in bed nowhere? Pantothenic acid is everywhere, but is numbness and tingling in the hands and feet nowhere? Pantothenic acid is everywhere, but is insomnia nowhere? Are muscle cramps, nausea, abdominal cramps nowhere? Is farting nowhere? Pantothenic acid should not be known as the B vitamin that you are least likely to become deficient in. It should be known as the B vitamin that we know the least about. This episode is brought to you by Ample. Ample is incredible. It's a meal in a bottle that takes a total of 2 minutes to prepare, consume, and clean up. 2 minutes. I'm not kidding. Now, I know what you're thinking. Anything that quick just has to be made of synthetic ingredients that you'd have a hard time pronouncing and wouldn't want to put into your body. But it's not. Ample is made entirely from natural ingredients and designed to provide an optimal balance between protein, fat, and carbs, as well as all the vitamins and minerals that you'd need in a single meal. There's no question that it's always best to sit down and take your time eating a home-cooked meal from fresh ingredients. But Let's face it, oftentimes we just don't have time for that. If you live a busy life like I do and your goal is to get things done, you need quality fuel that you can get into your system quickly. Here's a great example where Ample is perfect for me. When I shoot videos, it takes hours to set up and break down all of my equipment. So I try to get as many videos shot in a day as possible. This prevents wasting a lot of time on setup and helps me conserve big blocks of time outside of shooting videos to get into a flow state where I can research something to my heart's content and spend all the time I need thinking about it creatively and analytically. But if I spend hours dealing with recording equipment plus hours spent preparing food, eating it, and cleaning up, there's no time left over to actually shoot any videos. So on recording days, I use Ample to maximize efficiency and focus on getting things done. Ample comes in 3 versions: original, keto, and vegan. And each version comes in 2 portion sizes, 400 calorie and 600 calorie. The 600 calorie original version gives me 37 grams of protein from a mix of grass-fed whey and collagen, which promotes satiety and flips my brain on. Its fat comes from coconut oil and macadamia nut oil. I like these oils because they're low in polyunsaturated fatty acids, or PUFAs, oils that promote aging and are usually loaded into the processed foods that most people eat when they need something on the go. The coconut oil provides some medium-chain fats to keep my energy levels up, too. The carbs, the vitamins, and the minerals all come exclusively from food sources like sweet potatoes, bananas, cocoa powder, wheat and barley grass, and chlorella. It's full of natural prebiotic fibers and probiotics to promote a healthy microbiome, and the gentle sweetness comes from a mix of honey, monk fruit, and stevia. I just mix it with water, drink it, rinse out the bottle, and boom, 2 minutes in, and I'm fully fueled and ready to face the next phase of the day. I first came across Ample when I met its founder and CEO, Connor Young, at PaleoFX a few years ago. Connor inspired me with his vision for Ample, which I anticipate will be much more than a meal in a bottle in the near future. I've become an official advisor to Ample, and I'll be helping Ample design scientific research that will lead both to an ever-improving Ample and, I hope, meaningful contributions to our understanding of how to use nutrition to help people be healthier and happier and perform better at the challenges that they care most about. As a listener to the Mastering Nutrition podcast, I've also worked out a special deal for you. If you use the discount code CHRIS15, you'll get 15% off your first order of Ample. To get your discount, go to amplemeal.com. That's amplemeal.com, 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 and use the code CHRIS15 at checkout. This episode is brought to you by Ancestral Supplements. Traditional peoples, Native Americans, and early ancestral healers believed that eating the organs from a healthy animal would strengthen and support the health of the corresponding organ of the individual. For example, the traditional way of treating a person with a weak heart was to feed the person the heart of a healthy animal. Modern science makes sense of this. Heart is uniquely rich in coenzyme Q10, which supports heart health. The importance of eating organs, though, is much broader than simply matching the organ you eat to the organ you want to nourish. For example, natives of the Arctic had very limited access to plant foods and got their vitamin C from adrenal glands. And vitamin C is important to far more parts of your body than simply your adrenals. In his epic work, Nutrition and Physical Degeneration, Weston Price recorded a story of natives who cured blindness using eyeballs, which are very rich in vitamin A. But now that we understand vitamin A, we know that we can get even more vitamin A by eating liver, making liver good for your eyes. Our ancestors made liberal use of organ meats both to be economical and to utilize their healing and nourishing properties. Animals in the wild do the same. Weston Price had also recorded a story of how the zoos in his era were capturing lions, tigers, and leopards, oh my, only to watch them become infertile in captivity. Researchers then observed what the lions did when they killed zebras in the wild. What they did was they went straight for the organs and bone marrow, leaving the muscle meat behind for the birds. But even the birds took what they could of the organs and bone marrow. Price reported that once the zookeepers started feeding the animals organ meats, boom, their fertility returned. The problem I often encounter, though, is that many people just don't like eating organ meats. Let's face it, if you weren't raised on them, it can be very hard to acquire a taste for them. That is where Ancestral comes in. Ancestral Supplements has a nose-to-tail product line of grass-fed liver, organ meats, living collagen, bone marrow, and more, all in the convenience of a gelatin capsule. For more information or to buy any of their products, go to ancestralsupplements.com.
ancestral supplements, putting back in what the modern world has left out.
If you would like a transcript of this episode, you can sign up for the CMJ Masterpass at chrismasterjohnphd.com/masterpass/mastering nutrition. Using that URL or using the link in the description of this episode will earn you a 10% lifetime discount on that. You don't just get transcripts for this episode, you get them for all episodes of Mastering Nutrition and all episodes of Chris Masterjohn Light, which are short, quick, practical tips. And although Masterclass with Masterjohn is currently on a hiatus, which is detailed biochemistry lessons on specific systems in the body all arranged in a course, that will be coming back, and you do get access to the premium features on everything that has already been published in Masterclass with Masterjohn, and that does include lots of awesome features that include but go way beyond transcripts to help you learn the most out of the material. You also get the content early. You get it as it is produced instead of as it's released. And I don't promise exactly how much advanced that will be, but it's generally weeks and it's sometimes months in advance. Finally, if you sign up for the Masterpass, you also get a at least monthly opportunity to participate in a live Zoom Q&A where you can ask me anything about nutrition. Or if I have a guest, you can ask us about anything about a specific topic that intersects with nutrition. If you want the other episodes in this series on managing nutritional status, you can get them at chrismasterjohnphd.com/marker #episodes, or you can just go to chrismasterjohnphd.com/marker and scroll down to the episodes, or you can use the clickable link in the description of this episode. If you want an email update when the next episode comes out of Mastering Nutrition and when all the episodes of Mastering Nutrition come out, you can sign up at chrismasterjohnphd.com/mastering nutrition. If you want a dramatically more simple education on the nutrients than what you find in Mastering Nutrition, you can sign up for my beginner's course, Vitamins and Minerals 101, at chrismasterjohnphd.com/101. If you want to get all the super practical stuff about managing nutritional status for all the nutrients condensed down into one single comprehensive resource, you can get a copy of Testing Nutritional Status, the Ultimate Cheat Sheet at chrismasterjohnphd.com/cheat sheet, and you can use the code Mastering Nutrition to get $5 off. If you want more of my work in general, you can find me at chrismasterjohnphd.com. If you want more of Alex's work, you can find him at alexleaf.com. And you can find us on the internet. You can find both of us on Facebook, Instagram, and Twitter. You can find me on YouTube and every once in a while on Snapchat. All right, I hope you enjoyed this. Signing off, this is Chris Masterjohn of chrismasterjohnphd.com. This has been Mastering Nutrition, and I will see you in the next episode.